Hi, welcome to Take 15. Um, it is a lot harder to record this with me on the screen and thinking about what's happening behind me. Hopefully, my family is contained. Okay, I added some questions to the membrane PowerPoint. I really want you to try them, and then we'll. I'm going to give you the answers now. Give it a shot to answer the question. Check out what I'm telling you. Compare the two, and then any questions you have, please just drop them into the comments section on the Google Classroom and I can answer them from there. Pause this video at any time if you want to try the question first and then check your answer. Please know that I'm thinking about all of you. I really do miss you. I think the first week we were all thinking like, woohoo, break. And then now we're thinking this is weird and kind of unpleasant. I'll arrange a Zoom meeting for us at some point this week or at a few points this week just to check in and hang out. If you're actually trying to review stuff from the beginning of the year, you could really come back to school a total master of everything we've done if you spend some time working on it, if you have that mental, emotional capacity. So here we go. Let's look at these questions. That sounds very silly in there. So we're looking at a unicellular green algae that's photosynthetic. That information alone tells me that this organism has to be some kind of eukaryote. It's probably a protist, most algae are, but also it's truly photosynthetic and it's unicellular, which means that it can't be a plant. We genetically modified this organism by giving it a glucose transporter gene. This is important because you have to infer that this gene is going to make us a protein. Later on in the questions, there's a question about where you think that protein goes. Then we've got our graphs. Don't be intimidated. This is not scary. You can do this. I'm going to hope that home alone with all of the space and kind of your mental capacity clear for the next couple of days where you're not inundated with stuff all day, you can take a minute to think about this. Unmodified on the left, modified on the right. Our x-axis is time in days between 0 days and 8 days. And then cell density is just telling me how many cells I have in my culture. Remember, this is all taking place in a maybe a test tube this size. So 10 to the 7th cells per cubic centimeter. Cubic centimeter is the same as a milliliter. And then 10 to the 7th is going to be about 10 million. So 20 million, 40 million, 60 million, 80 million, 100 million, 120 million. That's a lot of cells. We then have our key over here. Filled in circles are cells in a light with no glucose in the medium. That's going to be our true control. That's how algae are. Think about ponds, think about the ocean. There's not just sugars floating about in there. There's no sugar in the soil outside. The plants have to make their own sugar by photosynthesis. But because we added this gene, right, this is what we're thinking about. So our next sample is light and glucose in the medium. So we're going to allow these cells to do photosynthesis and also feed them glucose. And our last sample, these are the cool ones. We're going to keep the cells in the dark, but we're going to feed them glucose. Let's find out what happens. First question, state the role of glucose in the metabolism of cells. The short answer is glucose is broken down for energy to make ATP. The long answer may be that glucose breaks down into pyruvate, that in the Krebs cycle, pyruvate is stripped of its electrons. Those electrons are then sent to the electron transport chain to power some proton pumps to make protons diffuse through ATP synthase to make ATP. Either way, glucose is broken down for energy, same way it is in you. So then we look at our two different types of cells, and we want to deduce where we would expect to find the glucose transporter protein in the modified algae cells. There are two ways you could go about this. One is just to sort of rationally think through the fact that if you're going to have a glucose transporter protein, the most sensible place for it to be located would be between where the glucose is and where the glucose isn't. So the outer membrane of the cell. The other thing you could do is look at the data here, especially these dark and in glucose. Cell density is a direct relationship to the cell's mitosis activity. So in these cells, with no ability to bring in glucose, no ability to do photosynthesis, we're not getting any new cells. In fact, most are probably dying off. But in this cell, where we've given them this gene for this transporter protein, even though we've put them in the dark, we've given them all the glucose they would probably, more than the glucose they would have made during photosynthesis, and we can see the population take off. So we know this right here is evidence that glucose is being moved into the cell, because in the dark there's no other way for the cells to get glucose, period. So this is evidence that the cells are going to have access to all the energy that they need and go ahead and reproduce. Because reproduction really is one of the first sort of priorities for most cells when they have energy. Carrying out their own metabolic processes is one thing, but trying to make more of their species is absolutely top priority for most species. All right, compare the effect of light on the modified and unmodified cells. This one has taken me a few tries to look at what data I want to explain to you. The first thing I looked at was light and no glucose, because this is sort of a control condition. And in both the unmodified and modified cells, we can see that it follows the same curve. You start with very few cells, you end up with about 40 million cells, give or take. This one follows the exact same curve and lands pretty much at the same point in the same way. The slight curve shapes difference can be accounted for just in individual diversity, but for the most part, with no other changes, we know that light allows cells to make some energy to do all of its metabolic processes, including reproduction. I then want to go back to the dark and the glucose, because again, these cells are having absolutely no opportunity to do photosynthesis, which means the only way for them to get glucose is to take it in. And the only way for them to reproduce is to have glucose. So in the unmodified cells, even though we've given them all the sugar they could want, they have no way to bring it into their cells. And in the dark, we know they're not doing photosynthesis. We know they're not making any, and their population stays real low. On the other hand, when we did give them the transport protein for the glucose and the glucose, we can see that their population skyrockets really fast. When we start at day 0, there's very few individuals. Here by day 3, we're already at the 20 million mark, whereas day 3, we're still pretty close to 0. There's a lot of nothing happening in the unmodified algae, but a whole lot of mitosis happening in the modified algae. And then obviously by day 8, we've got, hundreds of millions of organisms. So light provides the energy needed to manufacture the glucose, and the evidence we have is that in the absence of light, but with the sugar provided, the cells are able to reproduce. But in the dark, without the sugar provided, with sugar provided but no way to have it, you have no reproduction happening. We could also look at light and glucose. We'd say like, oh, okay, well, these follow each other, still no way to transport it, but plenty of light doing its photosynthetic thing. And then we can really separate these two, like, oh, they both have light, but like, this is the limit of how much glucose they could make. This is when we just throw it at them. All right, so here's the second part of these questions. Commercially unmodified algae are grown in shallow sunlit ponds or illuminated containers. The cells only grow in the top few centimeters. However, the modified algae can grow at any depth. So we want to think about depth in relation to light. You know that as you go down in the ocean, when you swim in the ocean, you really can't see your feet. Part of that is sediment, but part of that is just the way that light behaves when it hits water. Explain why the modified algae can grow at any depth, whereas unmodified algae can only grow on the surface. This right here is a direct relationship between the amount of sugar they're able to make. The unmodified algae can only grow at the surface because that's where they're going to be able to use the light to do photosynthesis, to make the glucose, to have the ATP, to do mitosis. On the other hand, the modified algae have access to all the sugar they could ever want. They'll still do photosynthesis if they have the light available, but if they don't, it no longer matters whether they can manufacture glucose because they're being fed it. So the modified algae can grow at any depth, at any density, and they'll still continue to reproduce because they don't need the light to make the glucose. They're being given the glucose. You ask yourself, why does this matter? This obviously doesn't happen in nature. You're right, it doesn't. But if we're looking at algae as a biofuel in the future, we have to think about how we can grow it quickly and efficiently. And if one of the ways is modifying it to have a transport protein so we can just feed it rather than waiting for photosynthesis to happen, that gives us an advantage on the technological side. State the two products of the light-dependent reactions of photosynthesis. So remember, in the light-independent, we got our oxygen coming out. We got our electrons. So coming out of the light-dependent reactions, we're going to have the vast majority of, out of the light-dependent reactions, light-dependent, sorry. Out of the light-dependent reactions, we're going to have oxygen as our main waste product. And we're going to have highly charged electrons. Those are our products. Our highly charged electrons are going to be added to NADP plus to reduce it to NADPH. So you could say technically the products are going to be oxygen and NADPH. Sorry, I read that wrong at first. I had independent stuck in my head. Oxygen and NADPH are what are coming out of the light-dependent reactions. Oxygen is kind of a waste gas, no big deal, but the NADPH is the thing, because remember, we charged up those electrons with the sunlight. We're going to take them to the light-independent reactions. We're going to bring in carbon dioxide. We're going to stick those electrons onto the carbons, and we're going to make our carbohydrates, our glucoses, for the future. So that's all for these questions. I hope everyone's okay. Again, please, please, please reach out if you need me. If you want to just chat in Google Chat or hang out on a Zoom conference just to have some human connection, I'm here for you. Talk to you very soon. Miss you a whole lot. Bye.